And likely you weren't satisfied with my explanation of C, and I wasn't either. So I ended up going online and trying to dig up and find some reference back to what's going on with that. And if you look at the slides, you'll notice there is now a subtle change in the slides. What is that subtle change? For the only two people that have the slides available, or if Mike's memory is that good. Subtle hint.
It's R, yeah, I was going to say that's R. Oh, never mind.
Previously it was referenced as the binding affinity, which is a nasty explanation, and that's ultimately coming back to Km. Because Km is a binding affinity, and we can't really define that according to our equilibrium dynamics, if you remember that kind of material from 152. Okay. C is the relationship between Kr and Kt. Morning. But we're meeting tomorrow.
Oh, tomorrow.
Yeah.
Got it.
Sorry.
Okay, no, that's fine.
Not a problem. Yeah, but this is where we will meet on Friday.
Okay, thank you.
Yeah. That was fun. I'm glad I recorded that. What were we talking about?
The dissociation constant.
Our dissociation constants. Those are legitimate dissociation constants. That is looking at how the enzyme in the relaxed form versus the taut form interacts with the substrate. Okay, so those are legitimate values. It's not this weird nebulous idea of Km, because Km has to factor in the dissociation to formation of products. These aren't looking at the formation of the product. These are looking at how the enzyme in either relaxed or taut state interact with the substrate. So there's no other kind of weird reaction coming out of those. Those are strictly dissociation constants. Okay.
RS is just relaxed state.
So,
State. It's not another.
No, RS, what is RS in this case? It's the substrate. So RS is the relaxed substrate complex. TS is the taut substrate complex. And these are our dissociation constants, okay, which again, kind of bizarre. So what is the dissociation? So let's pick, say R plus S goes to our RS. Well, our dissociation is the reaction coming apart. Okay, so when we calculate a K value, it's always products over reactants. Well, if we look at this equation, what's our product? RS or R plus S?
It can go both ways.
Depending on our perspective, it goes both ways. So this is where we have to be careful on what we're looking at with this K value. The K value in this case is the dissociation constant. It's referencing the dissociation, so your product is R and S over your RS. Same thing applies to your taut conformation. So the larger that K value, what does that mean?
The more dissociation there is.
The more dissociated it is. Okay? So if we're now comparing Kr to Kt, okay, R, we said, has a higher affinity for the enzyme, or for the substrate. Okay? If it has a higher affinity, what's its dissociation?
Lower.
Its dissociation value will be lower. So in all cases, when we're looking at our C, Kt will be massive,
So KT's big.
okay, and Kr will be small, okay, because it's not dissociating. What does that mean our standard C value is? Well, if we go and look at our C value, for our sigmoidal situation, Kt is big. It has virtually no affinity for the enzyme, or for the substrate. So we have a large concentration in our products, but we're dividing, which then means
Very small.
C gets very small. So when we look at our potential C values, we get C is 0.01.1. What happens to the curve as we get smaller and smaller? It becomes more sigmoidal. Okay? Which we did already reference before. The issue that we missed was looking at the next step of this, is now scaling that back in to understand this. Okay? And it all came down to what was the meaning of our Kr and Kt. That was the biggest issue. Okay? What happens if the binding affinity between the relaxed state, or the dissociation constant between the relaxed state and the taut state, become very, very similar.
Have more straight, right?
If our affinity between for the substrate, I keep saying affinity, if our dissociation between the relaxed state and the taut state becomes identical, there is no preference for our substrate. If there is no preference, what should our curve look like?
Line. Or
Careful.
just line.
Should look like a straight line. It should look like our Michaelis-Menten kinetics. Okay? So what's happening with our allosteric control is that Michaelis-Menten went through, they described the enzyme activity, but then we said, well, what the hell happens with this weird sigmoidal curve? There has to be some other factor incorporating into this. That's where we then reevaluate Km. If Kr and Kt are identical, that is effectively Km. Okay? That is saying we end up with that straight line right at the beginning of our curve, as opposed to that sigmoidal shape.
Because
Right.
KR and KT no longer play a part.
No
in
longer
the-
play a role because there's no difference between them. They're identical.
Gotcha.
Okay, which is when we would now shift to Km, which is why it gets more complicated when we look at Km, because Km is now looking at all those pieces. Kr and Kt are only looking at an individual state.
Because then it would be like a non-allosteric.
Exactly
Where
right.
there's
There's
no
no shape changing and
Okay.
all of that. And so depending on the enzyme, under certain circumstances, that change in shape is significant enough under inhibitors or activators, okay, that the relationship between Kr and Kt shifts enough that we see a sigmoidal relationship. Under some circumstances, those differences aren't enough and we fall into the Michaelis-Menten kinetics. Okay? So we're kind of approximating as best we can with the relationships we have. And Michaelis-Menten worked really, really well for certain enzymes. And that's because the relationship between the binding or the dissociation of the substrate with the relaxed form and the taut form were so identical that we couldn't see a difference. So we ended up assuming that was one unit. Okay? Some enzymes, there's enough of a difference between those relationships that we can then detect that, and that's how the sigmoidal curve starts picking up. Kind of make sense?
So KT is bigger.
So
sigmoidal.
KT is bigger, it is more sigmoidal, because we're saying that there's a bigger difference between the ability to hold on to the substrate. Okay, within our two states, relaxed and taut,
Because as it changes, and
it starts to shift more towards the relaxed form, which can bind substrate. That changes the binding constant.
they're both the same, it's more of the straight.
It's a straight curve because there's now no difference between the two.
Mhm.
There's nothing to really look at.
And then when KR is bigger.
So should KR ever be bigger?
It's just like
Well,
almost vertical.
KR is bigger, there is no taut form. There isn't really a situation that we're going to see that kind of, and I asked that same question and put it through my head. There's no situation where KR being bigger is going to change our kinetic system. We're going to end up looking at Michaelis-Menten kinetics.
Okay, so the only really matters is KT and as KT.
It's really only KT. So, and it's really the difference between them. KT needs to be larger than KR. Okay, if for any reason KT or that relationship starts to shrink, that's when we shift into Michaelis-Menten kinetics and everything's fine and, well, quote unquote, fine and dandy. Okay. Only when that relationship sets up where KT is significantly larger than KR do we start to see the sigmoidal aspect come in. Okay. So within our mechanisms, our concerted mechanisms are then saying, okay, well, what's happening? Okay, and we looked at our L, and I'm fairly confident that everybody understands the L aspect, right? That's just looking at the concentrations of those species. Okay, well, what happens to C? Okay, and this is where we got things confused, or rather, because I'm teaching it, I got things confused. And I already forgot what C was. KR over KT. Okay. As we add inhibitor, what happens to the binding affinity, or the dissociation constant for the enzyme with the substrate? Okay, it increases. Okay, it wants to dissociate even more because the inhibitor prevents the substrate from binding. Okay, due to that allosteric control. I know, it's kind of nasty. Our inhibitor binds, the inhibitor prevents, so it changes the structure and it prevents the substrate from binding. If the substrate is no longer binding, what happens to KT? Okay, KT was our dissociation constant, which was Taut times our substrate over our Taut substrate complex. Okay. What is our effective relationship when an inhibitor comes in? What happens to the concentration of the Taut substrate complex? That goes down drastically, which then means KT drastically increases. If KT increases, what happens to C? It gets even smaller, making our curve more sigmoidal. There's our relationship.
You gonna walk us through our test?
What?
So you're gonna walk, because that's what I'm gonna need, is you to sit here and walk me
And
through
so
this.
that's part of our aspects, and it's trying to tie all these things together. Okay? So yes, this is a valid test question to be able to look at all those relationships and nail all of those down. Okay? But is your entire test that topic? No. Okay? So will you see this question? Yes, because we're trying to decide, do people really understand what's happening within it? Okay, I have to ask that question. If you don't understand it, okay, you should lose points. Okay? How many points is that going to end up losing you? Is it going to prevent you from getting an A in the class? Almost undoubtedly no. Okay? May it prevent you from getting an A on this next exam? Yeah, that's a possibility. Okay? Because it's really getting at what are those relationships and what do they mean. Okay? So our KR and our KT are our dissociations. So what we have to decide is what happens as we put an inhibitor in, what happens as we put an activator in. How do those things change? Our L value, I think, made sense. That was just looking at our straight concentrations of taut versus relaxed. I'm fairly confident that you guys have that one nailed down. Should we walk through the L again? Okay, I heard no, we're sticking to it. Let's walk through the C once more. We talked about the inhibitor. Okay, so let's not bring in an inhibitor anymore. Let's look at an activator. What happens if an activator is now put in?
And then it wants to find more.
What happens, exactly right, if we add an activator, we aren't changing the taut form, we're changing the relaxed form now. So if we change our dissociation here, what happens to our relaxed form KR?
RS is gonna go up.
RS gets up, which means KR goes down.
Yeah,
Oh,
it
crap.
goes down, and then it's gonna make it even more, then the KR is gonna be a smaller number, but it's not gonna have as much.
But then C gets smaller. Dang it.
But KR is gonna be such a small factor, because you said KT is such,
But it's still, it's the difference between KR and KT.
What
There's our
did
problem.
go up?
KR is the concentration of RS. We're saying our RS concentration goes up. KR should get smaller as you bind activator, because
Right.
you're increasing the affinity for RS. So that concentration should go up. Your concentration of R and your substrate should theoretically both go down. So KR should get smaller.
wait, whoa,
But wouldn't-
go walk there again,
Well,
you just.
KT would also change too, wouldn't it, with an activator?
But does the activator interact with our taut form? So I think our activator situation makes things
No, but doesn't
a little bit
the
worse.
activator keeping the relaxed though?
So what happens as we add the activator? This value gets bigger. ARS gets bigger. What happens to TS? That one's a little bit weirder, but yeah, it's going to end up getting smaller. Okay, what happens to T? It's going to get smaller, and R gets
Bigger.
bigger.
Maybe it's proportionate so that C doesn't change.
So these values will counteract each other. And because TS was such a small concentration to begin with, that value going down isn't going to be by that much. Okay? Our R should kick up a little bit more. It still doesn't quite work. So what we're going to have to do is settle with this. Okay. As we change C, where's that curve? It's back here. Unfortunately, we're not going to be able to look at those direct relationships because I keep missing something, and it might just be with that definition of our dissociations. The bigger KT is, the more sigmoidal. Remember that aspect. We'll have to stick with that. Damn it, I thought I had it. Okay. That relationship is fair game. I won't drop in how does it change when you add inhibitor and all of that. I won't ask you about what happens to your C with the inhibitor or the activator. I will ask about L in the presence of our inhibitor or our activator.
So you said we're just looking at the relationship, the effect that happens to see with the change in KR or KT, that relationship?
Yeah. So if C gets bigger, we become more Michaelis-Menten. If it gets smaller, we get more sigmoidal. We get more allosteric. That relationship you need to know. Okay. And then ignore activator and inhibitor, how that's affecting C. Okay. Activator and inhibitor, I do want you to be able to describe how that's affecting our L, and that's looking at our concentrations of T and R. Okay, you guys should be able to nail that one down. Does that make sense? Sorry about that. I'll try. Actually, it's pretty easy to not let that happen again, believe it or not. The next one, as a mechanism, is the sequential allosteric enzyme. So what ends up happening is we get it kind of stepwise moving through as opposed to everything kind of happening almost independently of each other. So if we look at our initial state, where our enzyme is going to start in the total taut state, okay, not active. Substrate binds. As the substrate binds, that then causes half of our enzyme to say, ooh, I'm now binding enzyme, or binding substrate. I need to change my conformation. And by changing the conformation, it now moves to a higher affinity for the substrate, which then means we shift to the R form. That shift to the R form causes a structural change in the other half of our enzyme to change it from the taut to the R form.
Hemoglobin.
Like hemoglobin. Okay? That would be positive cooperativity. Okay. The negative cooperativity is happening in that, never mind, I thought I had positive and negative. The negative one is happening at the bottom. We have our taut form. The inhibitor binds, and now it goes, hell yeah, I'm totally locking this up, nothing's happening. We end up destroying any likelihood of substrate even potentially coming in, because the inhibitor binds and it changes the conformation to make it more of a taut form. Okay? So you get your positive and negative cooperativities in those situations. Okay? This works out kind of nicely because this matches then that induced fit model.
So what increases the cooperativity or whatever of the top form? It's just the substrate. No.
Well, so we could do activators as well, okay? But in this case, they're focusing on the substrate. Okay. Again, they're attempting to make it easier because we saw what happened in the previous case when we brought in an activator. It just turned it into a nightmare. So let's stick with that. Let's stick with the substrate. Theoretically, you're going to get a similar situation. Other questions? Okay, thank God. Now we don't have to worry about kinetics anymore. So phosphorylation as activation. So if we take a standard protein, so we got a nice image up at the top, looking at serine, we've got that OH. Okay. We can phosphorylate that by reacting it with ATP. ATP happens to be adenosine triphosphate. There's 3 phosphate groups. That last phosphate is relatively easy to exchange. Okay? So put in the proper circumstance, ATP can come in and drop a phosphate onto the serine. Okay? And we end up now with that phosphate group. How would that change how that enzyme or that protein could potentially interact?
So now there's 2 places to accept a hydrogen or something else on those 2 oxygens.
So why are you saying hydrogen? I agree with that.
Well, just because there's an alcohol there already.
Okay, so we have an alcohol in the first one.
Anything I should say could come and bind onto the last one.
What anything? So you said hydrogen, and I would actually step away from hydrogen.
Any kind
What's
of proton or positive.
positive? There it is.
Something.
Okay. The phosphate group is very ionic, meaning it's going to have a much higher affinity for water, be much more polar. It can now help to polarize different reactions. Okay. So we could phosphorylate, just put a phosphate onto our protein, which is now going to change the intermolecular forces and potential interactions for everything else. Does this mean it's going to make our reactions go faster? Well, if we're trying to do the reaction
The body,
in a membrane, let's say the reaction needs to happen in a membrane. Is this going to help the reaction or slow the reaction?
slowly.
It's going to slow it because what's in the membrane?
That's
Hydrophobic. Okay? So it doesn't necessarily make it go faster, okay, or slower. It's just going to change it. How it changes it kind of just depends on the situation that you're looking at. So we'd have to be a little bit more careful about nailing that down. So we're going to get some big allosteric changes here because ionic versus not ionic. Those big intermolecular force shift is going to cause the structure to change its shape. There's your allosterics. Okay. There's a fun question. What amino acids might we expect to be involved in this phosphorylation? Don't say serine. That's cheating.
amino acids like.
I'll kind of accept amino acids like serine.
Anything with a acidic hydrogen on the R group?
All of the.
It's not so much in the acidic hydrogen. It's actually the oxygen that's more important.
So it's gonna be
The ones that are polar uncharged.
Polar uncharged, anything with an oxygen really is going to be a really good amino acid for this interaction. Because if we go back and look at what's happening with our phosphate, okay, our ATP is something along these lines. It says ADP. Our oxygen can come in and attack that phosphorus. Those of you in organic chemistry, we would form a tetrahedral intermediate. Okay. That tetrahedral intermediate, our oxygen electrons come back down in a second step and kick out the ADP. And what we'd end up having done is now formed a new bond here between the oxygen and the phosphorus. So the oxygen of our protein is acting as a nucleophile or an electron donor. Perfect. Okay. So being able to recognize how our proteins are reacting or our amino acids are reacting in an individual situation is going to be important. So you need to know those definitions. Okay. Acids, bases, nucleophile, electrophile. When I reference acids and bases, I'm referring exclusively to how many types of acids and bases are there? I know that might be a stretch. There's Lewis.
Oh,
There's Bronsted-Lowry, and then those are the two I'd really only expect you to know. There's actually a third, Arrhenius. Arrhenius is a horrible definition, but it works for Gen Chem. Bronsted-Lowry, versus Lewis. Lewis applies to all reactions. That is way too broad. So don't refer to acids and bases according to Lewis' definition. We want to get a little bit more specific, and that's how I'll reference them. So if I say it acts as an acid, I'm referring to it acting as a Bronsted-Lowry acid. If I wanted it to act as a Lewis acid, then what I reference it as is an electrophile. Okay? Or a Lewis acid that is not a Bronsted-Lowry acid. So it's getting a little bit more particular with what is happening within your individual reaction. Okay? And those definitions are going to pop up here in just a second. The next big just kind of term, okay, because I know people like to harp on this kind of thing, kinase. Okay, it's just a nomenclature thing. You should know what a kinase is. They catalyze the transfer of phosphate groups. That's it. Okay, so if you see galactohydrase kinase, that's transferring a phosphate from some galacto piece to something else. Or it's potentially putting a phosphate on a galacto piece. So the name kind of gives you some information about what's happening within it. Zymogens. Zymogens are kind of neat because what it is the inactive form of our enzyme. Okay? So it's kind of like the Taut state. It's a little bit different than that. So one of the good examples is chymotrypsin. So if we take a look at our chymotrypsin at the beginning here in our figure, we're saying that that first place is the zymogen. Okay. So what trypsin does, or what another enzyme does, trypsin, comes in and it attacks the chymotrypsinogen, so the precursor to chymotrypsin, and it cleaves at certain residues. Okay, so it goes through and it cuts it open. As soon as it cuts it, the structure can now change its orientation. So if we now move to the next situation, notice that we have a gap in it. Okay. That gap now frees up the ability for this part to link back over to this part a little bit easier. In the process of now doing that, it changes its activity. Now it is partially active, and it can react with itself at 3 potential sites. So what ends up happening? It folds back on itself, and it facilitates the next cleavage. Notice that we're now missing some big sections out of here. By removing those sections, we now form the active enzyme. So this is kind of neat. We start with an inactive enzyme. An enzyme comes along, just does a simple cut, breaks it into 2 pieces. Those 2 pieces, through their side chain interactions, are still tied to each other through intermolecular forces. That subtle change in the structure allows it to then go back and bite its own tail and cleave a couple other sections. Once those sections are cleaved and then removed from the overall chain, it can continue to fold up to now form our uber protein that can now go on and do whatever it needs to do. Okay. When we talked about chymotrypsin, it cleaved at, it was tyrosine and phenylalanine, I think was our chymotrypsin. So now our alpha chymotrypsin is the full-on active form that can go out and start attacking all sorts of other pieces, okay, or other enzymes, proteins.
So chymotrypsin is the entire enzyme, okay?
The alpha chymotrypsin, so before we just said chymotrypsin, okay, it's really alpha chymotrypsin is the active enzyme. When it first gets synthesized, it gets synthesized as chymotrypsinogen. The inogen is in reference to the zymogen characteristic. So the zymogen is just saying that it's our enzyme, but it's not active. Okay? It's got all the proper pieces, but it's got some other stuff associated with it that's preventing it from acting. It needs a little post-processing. The post-processing comes in, does one simple cut.
So that anything will come in that will need to be deactivated that then
Yep.
does its thing. Okay.
Yep.
And that's how it changes from the ogen to
to the pi chymotrypsin, okay, it is now partially activated. It has some characteristics, but it's not fully activated.
And the alpha.
The alpha is now our full active enzyme. Yep. So it's just
What
kind of neat
brings
in
that?
referencing the name. What's that?
So what brings that? What brings the inactive, I mean, is it, there's lots of different things that would make it activate. We're not talking about that. We're just talking about what happens with this enzyme. Like, what makes it go from the inactive state to the active? 'Cause it's,
So to go from the inactive state, so number one, we have to synthesize the protein, which is going to be transcription to translation. Boom, now we got protein. But now that protein may not actually be the active form. And in this case, for chymotrypsin, when it's produced out of the translation process, it's not active. It's
is it
dead.
pH or is it like a?
It requires trypsin. I know that's confusing. We've
Okay.
got trypsin
So
and
trypsin is what comes in.
chymotrypsin.
So trypsin
So
comes
trypsin-
in and activates chymotrypsin to
Yep.
form the alpha chymotrypsin.
Yep.
Okay, so it's trypsin, okay.
So trypsin is the enzyme that triggers this. So why would we want this control? Okay, well, if we have a signal to form chymotrypsin, that could then form, but maybe it needs a second input. Okay, a second input to control whether we do this. Because what does chymotrypsin do? It starts cutting stuff up.
Is it fat
Okay.
or something?
It starts cutting up amino acids. It cleaved at the phenylalanine and tyrosine residues. Okay, at the carboxyl end, I believe, is what we said. Well, if it goes through and cuts proteins at those positions, do we really want a ton of chymotrypsin floating around at any given instant? No, because it would go through and destroy all of our proteins. Okay? So we only really want chymotrypsin active in case maybe, hey, we're running out of protein. We need some protein to build some other stuff. Okay? So start tearing down some old enzymes that aren't functional. So chymotrypsin, get to it and start cutting those apart so we can now work with it.
Why would it just tear into different substrates essentially?
So it would, exactly right. So it becomes problematic. All it's really targeting is tyrosine and phenylalanine. So any place that those are located, this thing's going to go in and start cutting it up. Okay?
Because I think it works in your digestive tract, so when you have excess protein, it has to break it down into amino acids because you can't absorb proteins unless they're
So
broken down.
a good location to put it would be in the digestive tract, because what do we want to happen in our digestive tract?
Break it down.
Break down all the food so we can get access to each of the individual parts. So this could be a really good place to dump this protein. Okay? But you up the concentration of it too much, even when you're not, if let's say you haven't eaten any food, what's it now going to start doing?
Digesting your mayonnaise, taking your.
Digesting everything else. Okay? So we do want to control its concentration. So that's what those individual triggers are pretty much doing for us.
So then does it have like a feedback inhibition type?
It's going to have some kind of feedback to shut it down.
Is trypsin part of the chymotrypsin? So
No,
is that-
separate enzyme.
Those two separate things.
Yeah.
So it's not as that one breaks down and the trypsin would feed
No.
back?
One extra thing to then add in here is this next fun thing. Just like we had kinases, we now also have proteases and caspases, which are ultimately the same thing. They're digestive enzymes. Their goal is to cut something up. Okay, cut it into smaller pieces. Okay. So, active site chemistry. What things should we be looking at the active site? Okay. Primary structure. Okay, that is kind of important. What is the individual sequence of amino acids? Not as important as, say, looking at the secondary structure. How does the sequence then fold on itself to generate different things? Okay. Organic mechanisms. How does it do it? How are the electrons moving to allow certain bonds to exchange places? Thank you. Finally, it happens. Okay, it does show up. The next issue is specificity. The specificity is going to come in when we look at our lovely topic of chirality, which my organic students would love to talk to you about now because they hate it. Okay? Nobody likes chirality because it's this weird, confusing topic that doesn't seem to make sense. How are my left hands and right hands possibly different from each other? But that's it. They have very similar functions, but they are different. That specificity comes in handy, no pun intended, when we're looking at our active site chemistry within our individual reactions. Okay? So, we already did look at this before, but we'll look at it again with our chymotrypsin. Okay? It cleaves your peptide at phenylalanine and tyrosine. Okay? So, primary structure, I need phenylalanine and tyrosine in my substrate. Okay? If they aren't there, can't do the cleavage. Okay? So that's kind of important. Okay? The next thing to also consider is, are there, instead of looking at the substrate, look at the enzyme. Okay? Is there some primary structure in the enzyme that's critical? So we're thinking about our amino acids. It's not just what the substrate looks like, but it's also what the enzyme looks like. Because when we call it an enzyme and substrate, in a lot of cases, they're both the same thing. They're both proteins. Okay? But one's acting on the other. Okay? So we have to be looking at both our substrate and the enzyme and deciding what things are important. Okay? So how do we decide? Go ahead.
Active site.
This is all kind of in the active site. Yep. Okay. So one of the things that we could go through and do is try and figure out what amino acids are important. Okay. And we vaguely talked about this earlier in the semester, and now we're getting at some kind of details to it. We can go through and tag certain amino acids and see how they affect the reaction. So there are a couple big tags that come in here. Right? Just because that one's easier to say, I'm trying to say this one too. We've got DIPF, which reacts with serine residues selectively, and we've got toothpick, which reacts selectively with histidine residues. So if we just take the protein and mix it with these reagents, anywhere there's a serine, DIPF is attached.
So those letters are
Are an abbreviation for the actual...
amino acids that are in this.
No. Toothpick and DIPF aren't amino acid sequences. Those are it's the abbreviated name for the structure. So if we looked at, yeah, there we go. I got one. Dichloromethane.
Okay, so it's actually talking to the specific.
What that specific molecule
makeup
is.
of-
Yeah. So when you see dichloromethane, we could refer to it as, and it very typically is referred to as, DCM. Because chemists are lazy and we don't want to write dichloromethane or even say it. So we say DCM.
And that's
Same
not
thing
part
is
of
happening
the
back up here.
Amino acids, that will be a separate.
It's a separate piece that we're then adding. That piece then selectively reacts with histidine in the case of toothpick or serine in the case of DIPF.
And those pieces that are part of the enzyme as opposed
Are
to
now
something
attached to
else.
the enzyme. Exactly right.
So an enzyme is a protein with other things on it.
So an enzyme can be just a protein. It can be a protein with metals coordinated in it. It can be a protein with different organic molecules coordinated within it. These we've actually put in there to label those positions, okay, to prevent the reaction. So what we did is labeled and said, oh, well, crap, it stopped working. What does that mean? That means those types of amino acids were pretty important to the activity of the enzyme. Okay? So what we ended up finding out by using these kind of labels is that serine 195, so the serine at position 195 in our amino acid sequence, and histidine 57, histidine at position 57, are critical to the activity of the enzyme. If we mess with them by adding one of these tags to them, the enzyme loses its reactivity and can no longer function. Okay,
So those were tags in this case to find out that those two
Exactly
amino
right.
acids were the ones that caused the other two.
Yep, exactly right. So an interesting feature there, 57 and 195, those aren't really close to each other in the primary sequence. Yet, they're critical to the chymotrypsin activity. What does that mean?
Could be close together, it's just that
They're probably
secondary or-
close to each other based on the secondary or tertiary structure. Okay. The protein is folding up and bringing those two amino acids close enough to be involved in the reaction. Okay.
So what are you showing in this yellow box? Like, what do you want us to get out of this yellow box?
So that yellow box is just showing what your different pieces are on labeling your enzyme. Is that what's going on here? No, actually, this is showing an example of the reaction working. So we're taking the enzyme. The enzyme sees this molecule, okay, which kind of looks similar to our phenylalanine or tyrosine. The enzyme goes in, forms a bond, kicks out our other piece, our substrate comparator molecule. Comparator. You at least understand what that means. It looks similar to our substrate. The enzyme then goes through and reacts with water to reform the enzyme, and it kicks out the other piece of our molecule. So what this box is just showing is that we are cleaving the bond in here. Okay. So it is, whoops, not there. I was going to say that didn't look right. There. Which is similar to the bond that we would be cleaving in the case of phenylalanine or tyrosine. Okay, in a standard substrate. So this is a substrate, what's the name? Substrate, oh, come on. How do I not have that name? Okay, I didn't put it up there. There's a specific word that I'm looking for. And all I can really say is it looks similar to the substrate. That's it. Okay. And if we'd used a standard substrate, your products would have been a lot messier. Here, what we can do is detect for the p-nitrophenolate ion and the acetate. Those are really easy pieces to identify.
So it's just showing.
So it allows us to then predict the enzyme reactivity or see the enzyme activity without having to look at a whole bunch of complex pieces that come out of it. So if we looked at our secondary, or this is really our tertiary structure of chymotrypsin, one of the things that's kind of interesting is if you look down in kind of the heart of that, thank you textbook for making it all black down there, so it's possible to see. This is histidine 57, and this is serine 195. Look at how close they are. They're darn close to each other. Okay.
So that's like the active site then?
That is likely our active site. And so once we go through and use techniques like X-ray crystallography, we can then start to nail down where these pieces are, see how they're connected, see how the structure folds up. Okay. In this process, we ended up also discovering that aspartate 102 is also pretty important, and that's what they're showing down underneath.
The 102, 57, and all that refers to its position in the string.
In the primary chain, yep, that's what those numbers are referring to. Okay. How do we know that is the active site?
Those are the two that.
Oh, because when you added the markers, it didn't work anymore.
Stop, shut it down.
We can put in our markers, and the markers can bind there. We could potentially use a substrate analog. There it is. Okay, a substrate analog that binds irreversibly in the active site. So it targets for the active site because it's looking for that bond to break, but then we set it up so that it can't break that out. Once it's now attached to the enzyme, we can then go through and set up crystallography. Get our image of the enzyme with this now substrate analog physically attached to the structure. We can then see that that's the active site because that's where it crystallized out. Okay? So, our mechanism, transition states. I will not ask you to do full-on mechanisms, and I may not ask you to go through and critique them, largely because our textbook does an awful job of describing them. Okay? So if we take a look at what's happening down in our active site, we're starting here, which they're labeling as the enzyme substrate complex. So our enzyme is this kind of green area. That's our enzyme. What's the orange area? The orange area is just open space. Okay. Your substrate is in here. There's your substrate as blue. So what ends up happening is the substrate makes it in through the open space into this pocket next to serine and histidine. Histidine was what type of amino acid? It was polar and basic. Histidine is polar basic. So what does it do? It shares its electrons with the hydrogen of our serine molecule. Okay? The hydrogen then has too many electrons, so the electrons in that bond need to go somewhere. Okay? Those will go and attack the carbonyl carbon of the amide backbone in our substrate. That then forces a tetrahedral intermediate. The pi bond breaks, and what we have is our tetrahedral intermediate, because if we look at the carbon where all our chemistry, primary chemistry happened, we have 1, 2, 3, 4 things attached, tetrahedral structure. Okay. The dotted lines are supposed to be showing weak interactions, okay, or hydrogen bonding interactions, not physical bonds. But because the enzyme is now kind of all tightly packed here, what can end up happening is that we will reform our pi bond, but then the carbon has too many bonds, so it has to kick a group out. The group it'll kick out will then be this nitrogen compound. Okay? That's the other half of the enzyme. And it'll do so by taking the electrons in that bond and sharing them with the hydrogen atom. But then the hydrogen atom has too many bonds, so the electrons go to the nitrogen of our histidine. So our histidine picked up a hydrogen and then waited a second and then dumped the hydrogen onto the other half of our leaving molecule. At this point, we've completed the first step of our enzyme cleavage, or we've completed the cleavage in our substrate. Here's our substrate 1 and substrate 2. Substrate 2 is connected to what? Nothing. So what can it do? It can leave. What about substrate 1? It is physically attached to serine in the enzyme. To get the enzyme back, I must cleave that bond. So while substrate 2 is leaving, water can come in. Water's a nice molecule because that oxygen can act as a nucleophile by sharing electrons. But it's not a very good nucleophile because it's not negatively charged. So we need to lose a hydrogen to be able to do that. So what happens? The basic histidine comes in again to help us out. Okay. It can help pick up that hydrogen, shuttle electrons to our oxygen. Our oxygen, now being a much better nucleophile, can attack the carbonyl carbon. Whoops. We end up with, again, a tetrahedral intermediate, because we've got 4 groups on that carbon. We go back down to reform our tetrahedral intermediate. Okay, the leaving group we want to kick out now will be up, whoops, that's an eraser, will be that oxygen. It's not a very good leaving group, so what it will do is pick up the hydrogen that histidine had picked up previously, and we now have serine off on its own as part of the enzyme, histidine off on its own, and we have substrate number 1. What is connected to substrate number 1? Nothing. It can now float off into the solution.
Is that other hydrogen still the one that's going to leave? Or is that a negative sign?
So this is one of the things that's kind of irritating within biochem, is they don't care so much about their arrow pushing and figuring out exactly what's happening. What functional group would we have there at the end?
It could be whatever other protein or stuff.
Nope, it's not whatever protein, because that is now cleaved. That's a functional group. It's a carboxylic acid. What did we say happens to carboxylic acids at neutral pH? They get deprotonated. So they're kind of dodging that bullet, or that question that you just asked, by sort of showing a bond to a hydrogen, which theoretically they just forgot to include. Okay. But at neutral pH, that's going to get deprotonated. Deprotonated by what? Anything remotely more basic than a carboxylic acid, which is anything. Everything is more basic than a carboxylic acid. Water would be able to deprotonate that. Okay? So that would be another step that's happening later on after the reaction has occurred. So what we're trying to do is look at how the electrons have shifted within the enzyme, how the enzyme reacted with our substrate, kicked off pieces, and shuttled things around. In each of those stages, what we end up looking at is how did our atoms react. So this comes to our definitions. Our Bronsted-Lowry acids and bases are proton donors and proton acceptors. The proton donor we've defined as our don't all jump in at once, our acid, and our proton acceptor, our Bronsted-Lowry base. If we go through and look at Lewis, okay, this is where I want to differentiate. Lewis looks at just electron motions. Bronsted-Lowry looks at proton motions. If you do an acid-base reaction according to Bronsted-Lowry, it is still a Lewis acid-base reaction. You just have to change your focus from the electrons to the protons, or protons to electrons. Okay? So if we're saying we are exchanging electrons and no hydrogens are involved, we don't even want to call it Lewis, because Lewis applies to everything. We need to come up with a new definition. That new definition is your electrophile and nucleophile. You're focusing on the electrons. Okay? And in particular, your electrons being donated and accepted, not involving hydrogen.
So in this case, we're looking at the electrons as they move around.
So if we move back to our previous example, let's look to see what happened in that very first case.
Okay, so what is the purpose of this? So the purpose
So-
was to attach histidine to serine with the hydrogen bond, like this whole thing was, that was what
So this
was.
whole purpose was to figure out how the enzyme worked to cleave that enzyme bond, or that substrate bond. Once we know how it works,
And how it
we
broke
can then
apart.
potentially change it to do something better.
Because we
for
wanted to
us.
separate, so that, the substrate, what we're trying to do is get that amine group to
So
bust off. and
we know
be
the
all
enzyme
itself,
does
and
this.
then-
Now what we're trying to
Okay.
do is describe it.
So I was like, oh gosh.
Okay. So let's look at each of those steps. So we've got 3 arrows in that first step. So we'll call it a red arrow, a blue arrow, and a purple arrow.
Do we be able to walk through this?
You should be able to do this, yes. What's happening in that red arrow?
The hydrogen is,
Nitrogen is donating a electron.
It's the nitrogen.
Okay. Nitrogen's donating electrons to what?
No, the hydrogen's donating
So what do our arrows show the motion of?
the nitrogen.
Arrows show the motion of electrons.
So maybe the oxygen is taking the electron from the hydrogen, and so the nitrogen is sharing proton with the hydrogen.
So this is one of the things why I dislike how biochemists reference things, is they aren't showing the appropriate steps. Really what we should be saying is in that red situation, we have 2 things happening. Nitrogen is sharing its electrons with hydrogen. Hydrogen then has to lose its electrons in that bond and send them to the oxygen. What happened?
So oxygen is more electronegative, so it's pulled them.
It took the electrons from the hydrogen, yep. Use our definitions though. Nitrogen acted as,
the electron donor.
so it did donate electrons, but what did it donate electrons to?
The
Hydrogen. So if we call that as an electron donor and then say, well, that's a nucleophile, Remember, our nucleophile said no hydrogen. We're sharing it with hydrogen, so we can't call it a nucleophile. What will we call it instead? It still donated electrons, that's fair. What did it donate electrons to?
Electron acceptor.
To hydrogen. The net result is what happened to the nitrogen. It lost electrons, so maybe it'll help if we look at what happened to the nitrogen in that next drawing.
It took where the hydrogen moved over to the nitrogen.
Yes, which means that
Accepts it.
it accepted a hydrogen. If it accepts hydrogen, it is,
A proton acceptor.
yep, what is a proton acceptor known as?
Base.
There it is.
Holy.
Okay. What does that mean our H acted as?
An acid.
Okay. We're really looking at the OH. This is now a proton donor, which is then a acid. What happened in the case of the blue arrow? The blue arrow started where?
The oxygen.
The oxygen.
Taken
Getting
electron
electron
from
out.
the carbon.
Okay, what did the arrow show the motion
No,
of?
it didn't take the electron. It gave me the
It gave the electrons. The oxygen is acting as an electron donor, which makes it a
acid. Base.
one of those definitions, yes.
Acid.
Keep your definitions together.
No, it's a base.
Okay, if we reference acids or bases, it is sharing or accepting hydrogen. That is an oxygen sharing electrons with carbon. Is that either of those atoms a hydrogen?
No,
So it is not acid or base.
so it's in a nucleophile.
It's sharing electrons. It is a nucleophile.
Holy crap.
What did the carbon act as?
Electron donor. Or acceptor.
An electron acceptor, because it had to accept electrons from the oxygen, which then makes it an electrophile. What's happening in our purple arrow? This one gets a bit interesting. Because when we look at our acids, bases, and nucleophiles, electrophiles, we're looking at two different species reacting. In the purple arrow, do we have two different species?
No.
They're attached to each other. So really calling it an electrophile or nucleophile isn't the best option. Okay. Why are we using the purple arrow? So the oxygen is for sure taking electrons from that carbon. Okay?
So the oxygen is the acceptor and the carbon is the donor.
So we could reference it as that, but did the carbon actually own those electrons? They were shared already between the oxygen and the carbon. So what is happening in that purple arrow? It's really balancing the actions of the red and blue, and ultimately just balancing the actions of the blue. Because if we didn't do that, how many bonds would the carbon then have? Too many. Okay, so the purple arrow is there only to really balance the actions of the blue. We could look potentially at this electron donor situation, but it's not the best option because those electrons weren't owned by any one atom. Okay? So every time you see curved arrows, this is what you should be thinking about. What did each of those species act as? If we're going to label a reaction as an acid-base reaction, the only thing that should have happened,
There has to be hydrogen involved.
exchange of hydrogen. What if you exchange hydrogen and do something else?
Anarchy.
Yeah. It's just
We'll go off our nucleophile, electrophile definitions. Okay? Because the exchange of hydrogen is really just there to help facilitate the other reaction. Okay? So when you're trying to classify reactions, okay, exchanging hydrogens, just acid-base. Everything else happening, you're looking at a covalent bond change. So we can refer to it as a covalent catalysis. Chymotrypsin, covalent catalysis. Why? We're changing the bonds between all of our atoms, or all of our enzyme and substrate, okay, that involve more than just hydrogen. Okay? Anarchy is not that far off. Okay? Questions about that? Okay. So, I do expect you to know these definitions, and I do expect you to be able to use them. Okay? So remember that. It is important. That is one of the things that drives all of the reactions we see. And when we get into, as you were actually even suggesting, I thought this class was going to look more at, systems, how a reaction works, Krebs cycle. It's all back to proton donors and proton acceptors and nucleophiles and electrophiles. Okay, so it will come back with a vengeance. Last part of whatever this chapter was, I think chapter 7, is they make reference to coenzymes. So these are things that are not part of the protein, but help the enzymatic process. Okay, so let's take a look at NAD plus NADH. Okay, so what we've got shown in that structure right below, the NAD plus NADH, is what we have here is NAD plus. Okay, all of that is NAD plus. Yeah. So what we're talking about is NAD plus would then go to become NADH. So what happened? How do we go from, oh, okay, I like that, saying we got a hydrogen. I want you to be really, really specific with this. What do you mean you got a hydrogen? What can you tell me about that hydrogen?
It was an electron donor.
It. Okay, so this is where we're getting two different definitions coming here. Why did you say an electron donor?
Because it was a positive, NAD was positive.
If NAD acted as a proton acceptor, okay, it would be becoming more positive because protons are positive. So what we are accepting here is actually H minus.
That makes
Okay.
sense.
H minus,
It's uncommon.
very uncommon because
No one's hanging out with oxygen.
tiny and negative. Tiny charges are bad, super, super reactive. So H minus is incredibly unstable. It cannot hold on to that negative charge. So what's happening is NAD plus is helping to remove H minus from a structure and putting it into itself and becoming NADH. This is facilitating a redox reaction. Okay. We are removing electrons from a particular place. Okay. If we take a look at our B6 vitamins, those are used to transfer amino groups. Okay. The structures may not be obvious on how they're doing it, and ultimately I don't really care. So don't stress too much on it, but that's what they're doing. Our NAD plus and NADH is going to become super important. Again, I keep picking on you, Mike. That's fine. For glycolysis, Krebs cycle, and all of that. Okay? So we need to be familiar with what's happening within that. Okay? It is really being involved. Did I not put it there? There it is. For your reduction and oxidations. NAD plus and NADH help with those types of reactions. How do you know a reaction was a reduction reaction?
Becomes more negative. I don't know.
Okay? Becomes more negative, but that assumes you know the oxidation state. What is the oxidation state for any atom in either of those two molecules? And long enough. So I agree with that. That's not a bad idea, but you took way too long to figure that out. So we need something else. Maybe try oxidation.
Reduction you gain and
electrons, but that assumes you know what the oxidation numbers are. Oxidation means you gain oxygens. Reduction means you lose oxygens. Pretty simple. Easiest way to remember it. Okay. There are other definitions or tweaks off of that, but that's probably the easiest way to remember it. The next big one is look at hydrogens. To oxidize something, you're putting more oxygens on it, which necessarily means you're losing hydrogens. Reduction is going to be the opposite, gaining hydrogens. So if you can nail down those two general definitions, that should get you through almost all of the redox kind of stuff that you'll see. Okay, at least in biochem. Okay.
What was that with hydrogen?
To oxidize something, you're increasing the oxygens on an atom.
Right.
Well, by increasing the oxygens, you're going to have to lose something else. You're going to lose hydrogen.
So redox is gaining hydrogen, oxidation is losing hydrogens.
Yes.
Okay.
Or gaining oxygen.
Or gaining oxygen, yeah. Usually you'll see them happening together. Okay? Awesome. Carbohydrates. So when we look at carbohydrates, they carry a general formula, CnH2nOn. Okay, where N varies from 3 to 8. Okay, so you've got a lot of possible structures that can come out of these. There are kind of exceptions to that. For instance, ribose matches this general formula. Okay, and we've seen, or you could see ribose in, say, RNA, ribonucleic acid. Okay, ribose also shows up in DNA. But in DNA, it's missing an oxygen. Why? What does the D stand for in DNA? Deoxy. You removed an oxygen. It's still a carbohydrate. It's still ribose. We've just removed an oxygen. So this is your general formula, your base formula. We can do some modification off of that and still maintain its carbohydrate nature, or its carbohydrate origin. The next part is we'll refer to them as aldoses or ketoses. Okay? What does ald or ket sound like? Exactly right. So depending on whether we are looking at an aldehyde as one of our oxygen-containing groups or a ketone as our oxygen-containing group within the carbohydrate, we'll refer to it as either an aldose or a ketose. So you need to be able to acknowledge what a ketone or aldehyde looks like. So one big tip-off, CHO is a very common abbreviation for aldehydes. Okay. Ketones you typically will not see abbreviated because there's not an easy way to do it. Okay, because it has to have a carbon off either end of the carbonyl. Okay? The polymer of one of these individual units then becomes our carbohydrate, okay, or a polysaccharide for many saccharides. Okay? So, simplest, monosaccharide, glyceraldehyde. Okay. Anybody notice some, or glyceraldehyde and dihydroxyacetone? Okay. Anybody notice anything interesting about either of those structures? You may not, that's fine, just curious. Highly water soluble, that is one of our characteristics of a lot of our saccharides, is they do dissolve in water pretty well. Lots of hydrogen bonds, which is why they would dissolve in water. It's a big hint. That carbon is tetrahedral. Not only is it tetrahedral, it has four different groups. It is chiral. Which means when we look at our simplest sugars, the ketone form, I don't have to worry about chirality. The glyceraldehyde, I do. So at my simplest monosaccharides, I don't have just two forms, I have three. Because I have each of the possible chiral forms, or the enantiomers of glyceraldehyde. And those structures are different, just like we saw with our proteins with RNS. Okay? Because glyceraldehyde is the simplest, that's the one that kind of started the nomenclature system that we'll then encounter when looking at these. Okay? We'll end up shifting to a Fisher projection. We won't look at these from the side or line angle anymore. What we'll end up doing is shifting into this newfangled little goofy presentation. So we've got the directions for us. Our highest priority carbon must be at the top of our structure. So within each of these, where is our highest priority carbon? So there's 1, 2, 3. Which of those is highest priority?
First one is double bonded to the oxygen.
This is our highest priority. That carbon must be oriented at the top. That's our aldehyde. Then carbon number 2 will go underneath it, and carbon number 3 will go underneath it. Okay. The carbon chain is by definition written so that it is always away from the viewer. Okay. With glyceraldehyde, that one's pretty straightforward because our carbon chain is really only looking at that point. So our view on this molecule is right here. We are looking at it that direction. Meaning the carbon chain is going away from the eyeball. So it's oriented exactly how we need to be. The bonds to the left and right then will necessarily be coming at the viewer. So we could show those as wedges. So we can add dashes in here. And we'll show these as wedges. From that first structure that I've drawn, where would that wedged OH be? On the right-hand side of your body or the left-hand side of the view? Yeah, that's exactly what you need to be doing.
On the right,
So if I could kind of levitate, I'd be levitating up above this molecule. My head would be up there, because that needs to be at the top. The OH is wedged coming out, so I'm levitating up. Where's that OH on? It's on my right-hand side, which means when I draw my structure, OH and H.
Ah, I see.
Okay? Because I've now defined all of that, I can then draw the structure instead as just this. And we get this just kind of cross. I don't need to go through, whoops, and do this kind of cross system for that bottom carbon, because that last carbon in the chain is not a chiral atom. So I'm only including chiral atoms in that cross notation. Okay. We could go through and assign RNS, okay, which I'm sure you would love to go through and do, and we'll actually avoid that, so you don't need to worry about it. But we'll introduce yet another nomenclature system similar to RNS, similar to plus and minus, similar to lowercase d and lowercase l, but entirely different, and you cannot say they're the same. We will introduce D and L, capital D and capital L. Okay. What those are referring to is the last chiral atom. The highest priority group on the last chiral atom is located where? It's the oxygen. The highest priority atom on the last chiral atom in our chain, from top to bottom, is located where? Right-hand side. Right in German is dextro. So this is the D form. If you went through and tried to draw, I'm going to have to erase everything, tried to draw that one, your result would end up being this. Where is the highest priority atom on the last chiral atom in this structure? So there's our last chiral.
yeah, chiral is the one near the center.
Yep.
Okay, that's what I was.
Where's our highest priority group? It's the oxygen. It's located on the left of the structure. So we will refer to that as the L form for glyceraldehyde.
So how does that one on the right side, how does that one have the,
I'm gonna ask you to
Alcohol on the
kind of
other
do
side.
that on your own. The dash means it's going away. So if you float in on top, it's on the left-hand side of your structure.
So that
Yeah.
chain was in the, or that glucose molecule was in the chain, in the middle of the chain, you would only look at the ending.
So here we want to take a look at some sugars. If we take a look at these, you'll love the names on this. This is known as erythros and threose. Okay? You notice we've added an extra carbon. You'll notice that they've already gone through and called these top two D. Well, how did they know it was D? Okay, we might say, well, the highest priority groups are on the right-hand side for erythros. But when you move to threose, they're on opposite sides. So why is it still D? Remember the rule for assigning D and L. The last chiral atom in your chain.
How do you pick which end of the chain?
So in our Fisher projection, the highest priority atom's at the top, lowest priority is at the bottom.
So this is the lowest.
You have to follow the Fisher projection. Your highest priority atom is on the right-hand side, D, erythros. Threose, same deal. How would we make these L and D? Or sorry, L for L erythros and L threose. Well, unfortunately, we've only set up erythros. You now have to swap it to the other side. Okay? Yes, massively confusing, but that's the system that we're working with. Okay? And we definitely have to stop there.